Well, first off, it's really great to be interviewing you today. And yeah, I mean, I was wondering if first we could start off with sort of you giving a quick summary of your research. I know you've done a lot of research, so this might be a little difficult, but yeah, just try your best to give the general highlights.
Our research, at least to summarize it, actually, I started already long ago, before you were born, in 1981, with research on DNA damage and repair. And at that time, of course, DNA was known to carry all the genetic information for very complex human beings or any other organism. And I thought, DNA must be a very valuable molecule because it contains, the primary information. And if that is damaged, yeah, then you can imagine that must have consequences. And that idea that has been fully borne out. So we started in 1981 when we didn't know anything but only the word of DNA repair. No, there was not a single gene that was isolated. There was nothing known about whether there are more than one repair system or whatever, and how many genes and how complex or simple. And the magnitude of the problem was not known either. So we worked on very rare diseases that are due to DNA repair deficiencies, where children are born with a defect in one of the repair genes. And we were the first in the world to be able to clone the first human DNA repair gene. That was the ESCC1 gene. At that time, we had some mutants that were repair deficient because they were very sensitive to sunlight, to UV, and UV damages DNA. That was just discovered. And these damages needed to be repaired, and these children are exquisitely sensitive to sunlight. And they also have skin cancer, more than 1,000-fold. So there was a link between a genetic defect, an environmental component, UV, and an outcome, cancer. So we started on that work in 1981. In 1983, we had the first repair gene, and we knew how to do it because it required a number of very strict steps that you have to check. Since we cloned half of the genes involved in nucleotide excision repair, that's one of the repair systems. Later it turned out there were many more, but that is an important one because it deals with a wide spectrum of damages. And we also clone many genes in transcription coupled repair, which is another complementary system. And that's really ingenious because the one is covering the whole genome from certain types of damages, recognizing and then eliminating a damage. And the other one takes care that transcription, when it is blocked by a damage, is rescued. Those two are very complementary systems. The one prevents damage that accumulates over the whole genome, and thereby, if it is not functioning, will lead to more mutations over the whole genome and lead to more cancer. Not so much early cell death, whereas the other deals only with the transcribed strand of an active gene. So the other part of the genome is not at all repaired by the system. Only when the polymerase cannot continue because it's blocked by a damage, then the system rescues. And so that is very important to allow a cell to survive the damage. Because if the transcription is blocked, yeah, you cannot use your genes, and then the cell will have a hard time, so will die earlier. So that last system protects from early cell death and also functional decline, because a cell can tolerate a certain number of lesions and not use the genes, but it still has another copy. Maybe the gene is not function, not important for vitality. So that one protects from cell death and from functional decline. And the other one, it primarily protects from mutations and cell death. So the one is against cancer and the other is against aging. So whenever we clone the genes, we invested a lot in trying to get mouse models for these genes, for these repair defects in humans. Actually, there are many patients and many diseases that were discovered subsequently. And sometimes we had the mouse and the gene before or later, there was a genetic defect that was linked with it. But and when we looked at the mice, then the mice, particularly in transcription coupled repair, and in retrospect, also the patients that belong to that defect, they showed many features of accelerated aging. So, you know, working on DNA damage and repair, we saw, oh gosh, these mice are aging. They became gray, they had kyphosis, they lived shorter, they had overall frailty. And the further we looked, the more we investigated, the more segmental, but not a full spectrum, but only a subset of organs and tissues that showed accelerated aging, but it's undeniable. Of course, the people in the aging community didn't know us, so they didn't believe the results. Because we could make repair. We had a single mouse mutant that had one repair defect, but we had many mice. So we invested the vast majority of all the mice in the world on those repair systems have been generated in Rotterdam in our lab. So we could cross these mice, and the more the repair defect, the double mutants, the stronger or faster the aging became. And so we had mice that lived, instead of 1 or 2.5, 2 to 2.5 years, that lived only 1.5 years with one repair defect, but that lived only 5 weeks if you made the defect worse by making a double mutant. So, and if we made even a mouse with 4 repair defects, and then it was not a full knockout because that would not be viable, but hypomorph mice, and they showed now the spectrum of aging became much wider. So instead of not only the post-mitotic tissues, the brain, the liver, the kidney, the skeleton, which are not much cell renewal, those depend very much on transcription coupled repair to keep the genome that they use for transcription clean from damage that otherwise constantly accumulates. So if you have a defect in transcription coupled repair, then the aging is primarily focused towards the tissues that were not dividing so much. But if you are constantly dividing or regularly dividing, you dilute also the damage that has accumulated in the meanwhile. And you allow repair to take place while the replication machinery encounters the damage. So replication dilutes and allows repair of accumulating damage. Whereas if you do not replicate, like the neurons, they have to live for maybe 1 century, they have to constantly, clean the genome, at least the part of the genome they use for their function, from damage that accumulates. So then we decided, we saw all-round aging in mice that have repair defects important for post-mitotic tissues, and other repair defects in other pathways that are very important for the proliferative compartment, the hemopoietic system, but, for instance. So we started to, first of all, we saw a causal relationship with DNA damage repair. And the stronger the repair defect, the faster the repair, the aging. So there is a dose-response relationship which indicates it's causal. It must be the DNA damage that causes this. So in the beginning, in 2004, when we published a paper in Science that, we had mice that could age in 5 weeks because they had a very severe repair defect. And by the way, there are also children that live only a couple of days or weeks and then die from many problems, but nobody knew that actually the aging was interfering with the development. So, but at that time, the community in aging, wow, no, they cannot be true. So they published a satirical poem, Snippety, Dr. Jan Hoeijmakers, that's me, tired of working with slowly aging beasts, the mice age slowly. Lops out 1-deoxyribonucleotide, now his mice age as quickly as yeasts. Mice that age as fast as yeast, oh no, forget it. No way. No. It's absolutely true. And now the field is more and more, be convinced that DNA damage is very important. And we think it's the main cause of systemic aging, systemic. So for for the whole body. And, if you have a mutation in ApoE, or if you have a mutation in beta amyloid, you may have accelerated aging in one or a few organs. But the systemic component of aging, we think, is really absolutely due to DNA damage that interferes with transcription, particularly in the post-mitotic tissues, and that interferes with replication, particularly in the proliferative tissues. And that causes what we think the systemic component of aging. And the evidence is so, if you put it all together, is so completely convincing, there is no escape. And I'm always very critical on particularly our own findings, because the last thing I want to have is that somebody else finds out that we were chasing the red horse, or that we were on a wrong track. So so we have mice that in many different kinds of repair deficiencies that all show unequivocal a process that is in no way different from the normal process of age, except that it's faster and more exaggerated. So for instance, in mice, the neuronal system is not so, in a normal mouse, a lab mouse, the neuronal system is not showing a lot of aging. There is, of course, there is some, but there is no Alzheimer mouse. As far as I know, no spontaneous mouse in Alzheimer. Well, our mice, I'm absolutely convinced, and we are now working on it, are the best models for Alzheimer and Parkinson, because the most important risk factor for all of these diseases is your age. So Alzheimer, Parkinson, frontotemporal dementia, Huntington, ALS, they are all aging related. And you see all kinds of proteins aggregating. And we understand now, it's not yet published, but we understand fully why proteins start aggregating in neurons when those neurons start aging.
Wow.
Starting with a DNA repair defect, starting with a DNA damage problem, leading to transcription being affected, leading to a protein imbalance, leading to proteostatic stress, and then proteins that normally can be kept under control by the proteostasis machinery are gradually escaping, and they accumulate because they cannot be degraded. And that's why we think all these proteinopathies arise. And in one person, it may be the beta amyloid gene, because that one is more expressed in the cells, or it has more tendency to aggregate when you have a mutation. So then that becomes a limiting factor, and then you get that phenomenon of aging much earlier. But the phenomenon that it occurs, I think we understand. And then we found something else that was also very informative. So we had mice that show accelerated aging everywhere, or mice primarily in the liver or the kidney, depending on which repair defect. We have even, with gene inactivation targeted only the cerebellum for accelerated aging, or the vascular system, or the liver, or now the Laura Niederenhofer just published in Nature an article in the immune system where you see immunosenescence, and that links also with corona, by the way. But anyway, so we can target aging to any organ, but when we study aging in these organs and tissues of these mice, with gene, microarray analysis and next-gen sequencing now, we saw immediately, I saw it, I was behind the computer when the first readout came from the microarray analysis together with my colleagues, and we saw one of the top genes was the IGF-1 gene going down in an organism that is aging too fast. So we could say, if you have too much damage, then the system responds by suppressing growth. You have to suppress your growth. You don't want to waste energy and resources to growing. You want to try to protect yourself. So the antioxidant defenses were all very strongly upregulated. So instead of growing, these mice, and the children, by the way, too, which have the defect, they stay very small because they are constantly prioritizing their maintenance systems, their defense mechanisms to become resilient to accelerated aging. They, you know, if you're young, you don't want to age too fast. So then the system, oh, stop. I have to try to prioritize to protect myself. And that response, we saw it immediately, resembles dietary restriction. Dietary restriction is an anti-aging intervention that in yeast, in worms, in flies, in non-human primates, delays aging. If you have too little food, then the body wisely does not waste it to growth. Now it invests it into maintenance to try to survive the period of starvation. So we call this a survival response. And that triggered something very counterintuitively. So we first discovered this connection with, so that mice that show accelerated aging have a response that resembles delayed aging, so the opposite. So you know, yeah, but what would happen if we would take these small mice that live short because of aging, if we put them on a diet? The last thing you want to do with a child that is, you know, 5 years and still, you know, very small, is to put it on a diet. These children get all extra food because they have to grow. So we applied dietary restriction to our mice, and then, and you know, it was unbelievable. These mice lived 3 times as long. So instead of dying we started from week 7, put them on a 30% dietary restriction. Then normally they would live about 19 weeks. They live about, that mouse lives about half a year at maximum, 26 weeks, 27 weeks. Now it lived more than a year. Wow.
Yeah, wow.
Less food and you live 3 times as long, and they were in a much better shape. And particularly neurologically, they were doing much better. So then I said, oh, wow. So nutrition and the amount of food, it's the calories particularly, that determines, you know, whether you invest into growth. If you're doing well, then the body knows, okay, now I have to invest into growth because you have to become an adult. And of course, give progeny. And then if the progeny is independent and can take care for itself, then of course, your biological function is over. So we said, okay, so you can use your energy for 2 purposes. In case there is reasons, in the case of a crisis, then you can still, you can try to protect yourself, to try to survive the period of starvation, or when you are very ill. So when you have high fever, you do not have much appetite. Because the body at that time doesn't need food. No. It wants to try to survive, to fight back against the infection. And everybody, when he or she is ill, your appetite is disappearing. And these children that stayed small because they had a repair defect and aged too fast, they had no appetite. So these children all get extra food by tube feeding or directly into the stomach. So we said at the meetings with families with these children, you know, yeah, our mice, they indicate these children should get less food instead of more. I gave 3 years after, behind each other, again a presentation, and I show again more and more data that showed that these mice were doing much better, in a much better health neurologically, completely, you know, The mice that were on a diet, they could run. And the ones that were eating all the time as much as they liked, they hardly could walk. They had tremors. They couldn't keep balance. They had spastic contractions. They resemble, to some extent, Parkinson. So food is very critical for these children. So, but nobody did anything. So then I, started to say to clinicians, we should try this in children. And I was accused of criminal activities. But now the first child has done it. And the results are astonishing, really astonishing. So that was a child that had a disease which is called Trichothiodystrophy. They are very rare diseases and they are very, 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 very severe. And no medication, there's nothing that we can do. And this child was 6. These children, it depends on the mutation, but many don't get older than 5 or 7 or 8. And she had tremors. She couldn't put 2 Duplo blocks on top of each other because she was shaking so much. Very bad sign, because then they get paresis, so they become paralyzed and they die from neurodegeneration. It's very, very tragic. So I had already talked with the parents and their doctor believed our results. I know him and he knows that these results are reliable. So together with the parents, we decided this child got 1,150 kilocalories every day directly in the stomach because she had 0 appetite. I have seen the parents struggling to get 1 spoon into her mouth. And she had no appetite. She would spit it out again. So she got 1,150 kilocalories directly in the stomach. So we said, no, if we gradually decline every day or a couple of days, 100 kilocalories less, and see how she responds, and then continue. So within 12 days, she lost her tremors. She could put the Duplo blocks on top of each other. She had never in her life, even, these children can never walk. Or if they can walk, then, it will be finished in a couple of years. And then she had never spoken. She couldn't pronounce words. Reading and writing are totally impossible, unthinkable even. She started to walk independently. And now she bicycles. And she is 3 years, stable health. Less food. And now the whole guidelines for these diseases have been completely reversed. Instead of more, these children should get less food. I would be able to show you movies, but it is not yet published. We are writing up a manuscript. It shows the value of trying to understand what is going on. And we came from a very different angle, from the field of DNA damage and repair. At that moment, nobody had made any link with aging. With cancer, yes, but not with aging. Now we are convinced that this is the main cause of systemic aging. We just published a review in Nature on the central role that DNA damage plays in, we think, systemic aging. And we have also found that for these children, the amount of food is extremely critical. And these children over-respond to less food. With normal aging, you also benefit from dietary restriction in all organisms. That has been shown, not to the same extent, because these animals are very sensitive to the damage, much more than you and me are, who are repair competent, who are proficient in our repair systems. But these children are not. So they are more than we are sensitive to damage. And with less food, your metabolism is redesigned and you generate less reactive actin species, and you have better protections, and other metabolic changes take place. And so you have less damage. And these neurons that were causing tremors because they were not functioning well anymore, that they couldn't control the muscles because they were already, you know, declining. In fact, they had not yet died. So these neurons, what we think is they had now less damage, so they could better cope with the damage and then function better and then get again the muscles under control and balance. And she could speak now for the first time. Wow. She started to speak. And for the first time, she could count. And for the first time, she started to write.
That's amazing, yeah.
A child at 6, that was, I would have guessed, but that's of course, we don't know. But many of these children don't get older than 5, 7, 8. I don't know about this child, of course, but now she is now over 10. 10 and what is it? Oh, it's not, yeah, one day. So 5 months, 3 years and 5 months after the period that she started the dietary restriction, and she is doing very well.
That's amazing, yeah.
So, and other children, what I hear from parents, they are also very positive. So yeah, I think from an unexpected perspective, but I think this is really, yeah, it shows how science and the scientific journey can sometimes take you in unexpected directions.
Yeah, completely. That's incredible.
And then all of a sudden now we understand, we think Alzheimer. Because these children, they also have many features of Alzheimer, even though some of them die by the age of 12 with another disease, which is called Cockayne syndrome. There are tangles that are already found in the brains of these children at the age of 12 years. We think they are representing the main risk factor for Alzheimer and Parkinson aging. And we can also, direct that accelerated aging to the cerebellum or to the hippocampus. We have done that, or to the forebrain or the heart. Yeah, I think, that is a summary, but it is a very extensive summary. Sorry, but
no, okay.
But it was a struggle, but with a satisfactory outcome. And I hope it will also lead in the end to clinical benefit. And for these children, for sure. But we think for many other areas in medicine too.
Yeah, completely.
So even though I retired, I reached retirement age 3 or 4 years ago, I am expanding in other labs. And we would like to translate what we found in basic fundamental research more into the clinic in different directions.
Wow, that is absolutely fascinating. Thank you for that summary. I'm sure everyone who's watching this will be really interested in hearing all of that. That was really interesting to listen to.
Yeah, it is in different pieces in literature, but to have the complete picture, that takes some time to grasp it.
Completely, but that's so cool, and it's really interesting. I mean, kind of when listening to that, I came up with a few questions in my head. I have a few pre-planned ones, but I'll sort of go with one of the ones I sort of came up with when listening. But I know you mentioned sort of DNA damage and DNA damage repair in its relation to a lot of the other aging hallmarks. But I was wondering, sort of, do you think generally DNA damage and DNA generally repair sort of causes these other aging hallmarks, or do you think sort of it's, I don't know, do you think it's the root cause for sort of the, I guess, other 9 aging hallmarks we're seeing?
Yeah, I think so. So what we now see, I'm now working also in a pediatric oncology hospital in the Netherlands. And children which when they are young, unfortunately get cancer, Fortunately now, these days, 80% survives, and that's because most of them have kind of leukemia, which we can cure because of chemotherapy. Chemotherapy that damages your DNA, the DNA of the tumor, and then the tumor dies, and you are cured. But it causes also damage in the body in other places. And what we now see, and what we predicted before it was found, that all of these children show accelerated aging in all organs and tissues. The more we investigate, the more we find. So chemotherapy that damages your DNA throughout because it's given systemically. 10 years later, these children are 20 years older. And so it shows what DNA damage causes in terms of aging, and it is aging everywhere. And we see, as a secondary consequence, we see that many cellular processes get affected. So for instance, proteostatic stress, we can fully explain, starting with DNA damage and transcription and protein synthesis imbalances, incomplete protein complexes, protein degradation machinery that comes overwhelmed, and itself gets also affected by this problem. So then you get proteostatic stress. So proteostatic stress is a secondary, tertiary consequence of a problem that starts with DNA damage. The insulin growth factor response that suppresses IGF and growth hormone all down starts with DNA damage. If you damage a cell, certainly if the damage interferes with transcription and is persistent, then even cells at a single cell level, you get suppression of the IGF. So this nutrient sensing is all caused by DNA damage. We can, and we think now we understand why, because the IGF-1 gene is a very long gene and that captures more damage. So we think now that this gene is purposely long. It's a small protein of about 150 or so amino acids, but the gene is 85 kilobase.
Wow.
This is for a short gene, for a short protein, a quite long gene. But with aging, you accumulate damage. The damage leads to less transcription. Less transcription leads to less IGF. So when you get older, IGF goes down. Simply because DNA damage more and more interferes with transcription of this gene.
Wow.
So the nutrient sensing pathway, we can completely understand why that goes down with aging, and why, if you do it, if you do it genetically, you boost your defense mechanisms, you suppress your growth, and you last longer because you have more maintenance, and so your genes get less damaged, and there is more repair probably. That's what we do not know yet, but so you protect yourself better, and you last longer. You stay longer, healthier. You don't grow that well, but you stay longer. So, but also telomere attrition is actually DNA damage, because if the telomeres are too short, then there are breaks, and breaks block replication, and signal immediately stop cell cycle. And cellular senescence is one of the results. So cellular senescence in nature, I think most of the time comes from DNA damage that accumulates. And if you treat an organism with doxycycline or doxorubicin, I mean, or if you cisplatin or so, then you see a senescence response because they cause damage. So senescence is also mitochondrial DNA and mitochondrial function. Mitochondria have DNA damage too. So and repair is not so well investigated in mitochondria, but also more than 1,000 genes for mitochondria are in the nucleus. So, and they get damaged too. So that mitochondrial function is affected by DNA damage it's very logical. So all of these hallmarks, we think in terms of hierarchy, they may well involve and start with a DNA damage problem to begin with. And DNA is the only molecule that you cannot make from scratch. You have to, you can only repair it. So all the other components, RNA, protein, membranes, organelles, you can renew in the cell, but DNA, is the one that contains all that information, but itself not. So that also, in that respect, makes some sense.
Yeah, completely. That's really, really interesting. And it's also cool to see sort of how, I guess, the flow charts will work for this theory, because, sort of DNA damage, I guess, is at the, the root cause in this theory, but like then, it's all the sort of other, all the other aging hallmarks sort of come from that, which is fascinating. So yeah, that makes sense.
That's what we think now. And I think although of course not in all cases the evidence is completely conclusive, but everything points in that direction and is consistent with what we know.
Yeah, completely. I mean, it makes a lot of sense. I sort of, this is a bit more of a general question and not necessarily around, the science itself, but I was wondering what is the most important thing you think we can do right now to create good progression in the field of longevity and aging?
You mean which disease?
No, I mean, just like in general, like where could we focus our efforts as a field? I mean, honestly, I guess sort of to rephrase the question almost, like, what sort of obstacles do you sort of have to overcome right now within longevity to sort of have more progression as a field, and maybe not even longevity, but just aging too? Like,
I think, yeah. There are a number of important considerations to, so I think one of the fundamental problems is like the neurodegeneration. Even though we can replace in many organs and tissues the cells that die by new ones, and maybe we are able to improve this in an organism. Every neuron that you lost, every post-mitotic neuron that you lose in the course of your life is gone. And all of its complex connections with all the other neurons and its function in a network. There is not a new cell that has all that history. So neurodegeneration will remain a very fundamental problem. And that will be a challenge for the future. Of course, senolytics maybe for some tissues at some moment in time be a solution, which I'm not working on it myself, so I cannot fully say, but what we can say for sure that every cell that you lose, either you lead to organism and tissue atrophy, or you have to replace it with a new one. So then the stem cells have to be in sufficient condition. Otherwise, you have to, cell have to divide to replace the one that has died because of a senolytic cleaning of that organ. Well, that will also raise risk for cancer. So there is always a balance between the two. So I think there is promise in senolytic agents, but maybe not for all organs and tissues, and certainly not in the brain. If all the cells that are not doing well and are on a senescence trajectory, if you get rid of them, then you may lose your memory, or you may lose certain neuronal function. So that would not be the best solution. So I think we have to still learn more about it. And of course, knowing what nutrition does and how we can trigger this protective survival response that invests more in the resilience, in the maintenance, in defense mechanisms, in the immune system. If we would be able to trigger that artificially, that would be, of course, a major benefit, because then we will delay aging in the whole body. In the brain, and particularly the neurons have an enormous benefit from this. That's what we find in our mice. So that would be a priority, because dietary restriction is not very popular, because we are always in evolution selected on whenever there is food to overeat, because maybe tomorrow, when there were no supermarkets and refrigerators, maybe tomorrow you wouldn't have food. So you have to build up reserves. But now we have constantly plenty of food. So we are constantly overeating, because maybe tomorrow there wouldn't be food, but that is not very good. because then our systems are thinking everything is okay. I can, with it going this way, I will become an adult and I will give progeny without problems. Why would I overinvest in maintenance? That costs energy, and it would go at the expense of progeny. So the system is very much focused. If there is food, then take more than you maybe now need, because tomorrow there may. So we are constantly, and that is something that we have to try to find effective solutions for, that prevent us from constantly being hungry.
No, that makes total sense, and that's super interesting too when you think about it. Sort of how our situations have changed since the beginning of time, and how sort of our habits from then might not be so good now because we have just completely different situations, and we do have more access to food. So yeah, no, I completely agree. And another thing, sort of just another question I had was, this is more just general advice around longevity, but sort of what is sort of advice you have for people who are younger and want to break into the longevity field? People who are younger.
To make choices in the field for your scientific directions.
Yeah, so basically.
You. Sorry, I didn't hear you.
Oh, sorry, I was muted. Yeah, I mean more in the sense of sort of what can you do now to sort of almost get into the longevity field? How do you yeah, I mean even in high school, just maybe tailoring it a little bit more. What classes should you take? Sort of how do you educate yourself in the longevity field when you want to sort of break in and you're young?
So yeah, I didn't fully understand it because the connection is not that good here. But what I for young people, children at school or students at university, I think biology, the direction of biology keeps so much promise for the future. Now we are more and more able to understand important biological processes and also to influence them. And this is the only sensible way to try and see whether we can improve our health, prevent diseases more than curing, but prevent them in, if possible, lifestyle, but in other ways, with medication or with early screening or in whatever way to improve quality of life and well-being in the end. And I think that I would certainly advise anybody, if you think this is something for your life to accomplish, then that would be a direction that I would take, and certainly, not only take, biology in the broadest sense. I mean, artificial intelligence can be very helpful in identifying, powerful. So bioinformatics, extremely important. We have, found it ourselves. And move every time, your development in your field requires another level of another discipline, another level of understanding. So we started with cloning genes, and then understanding pathways, and then making mice, and then looking at all the organs and aging in different organs and tissues, and now with nutrition, and then now with clinical trials. We are four clinical trials are now ongoing on the basis of this research. So every time it's a new level and requires new skills and new collaborations. Collaboration is extremely important. I cannot do everything, and certainly not, a team cannot do everything either. So you have to work and put the people together in a right configuration and with the right attitude, the right mentality, the right, solid, we want to do it together jointly. Then you can really make breakthroughs in the field. And that is what I would certainly advise.
Yeah, thank you so much. That was super useful. And that's really good to know about how, I mean, I completely agree. Biology is fascinating. And, there's such a direction for it, obviously, as it intersects with aging very much. So that's really cool. And yeah, I mean, overall, those were sort of the main questions I wanted to touch on today. And yeah, I mean, thank you so much for giving that sort of really interesting description of your research and answering all those questions so well. it was really cool to interview you today.
Thank you.